Chapter 5, Eukaryotic cells. And this is just a concept check if we're talking about eukaryotic cells, which we have a little bit, we talked a little bit about it before. Which part is responsible to coat from outside? The glycocalyx. These are basically the organelles inside the eukaryotes. Remember, we talked about, in the previous chapter, we talked about the difference between prokaryotes and eukaryotes, right? And we mentioned that the most important of all is that the prokaryotes do not have a well-defined walled or a nucleus that have a membrane, right? While in the eukaryotes, it is a well-developed nucleus that have a membrane. On the other hand, the eukaryotes have all these different types of organelles that's within a membrane, most of it within a membrane, while the prokaryotes does not have everything. Okay, so this is basically, if you look at it, it just looks like our cells, the cells that we all know, nucleus, nucleolus, endoplasmic reticulum, and so on. We will go through a little bit of details, but it's the same thing that we did in physiology before, in A&P. So nucleus, obviously, this is in the center of the cell. This is the manager of the cell. This is the part that contain the chromosomes, the genetic material, DNA, and so on. Nucleolus is like the nucleus of the nucleus. It's a small organelle within the nucleus. And the function of that, if you still remember from A&P, it's just to help us to make ribosomes and ribosomal RNA. Ribosomes and ribosomal RNA. This is a nucleolus. Of course, you can be asked about this, each one of those. And we will discuss some more details. Endoplasmic reticulum, we talked about this in A&P. There is rough and smooth, if you still remember. The rough ones in the plasmic reticulum is the one that have ribosomes on the surface, while the smooth ones does not have ribosomes on the surface. The one that does not have is to make lipids, and in both of them it's for transport. Golgi apparatus, just like our Golgi apparatus, to package, modify the products, specifically proteins, prior to secretion. If you wanted to secrete something, we called it in A&P, we call it the finishing deck, if you remember. We like if this is a factory, you're making anything, right? We're making TVs, for example. We're making laptops. You don't take it from the machines directly to outside, right? You have to send it to the finishing deck. They take care of it. They make sure that everything looks fine. They paint it. They package it, right? They put it in a good package, and then they export it to the outside. So this is basically the Golgi apparatus. It take it, it pack it, it modify it, and so on. Lysosomes. Lyso means lytic, which means to break down or to digest. Somes means bodies. So lysosomes are the bodies for breaking down, which is basically digestion. Digestion of what? Anything. You can digest anything. Any organic material can be broken down in these lysosomes. Vacuoles, this is something that we didn't talk about in our physiology, in human physiology. Vacuoles, or there is something similar to that, but we didn't call it vacuoles. Vacuoles are basically cysts that we temporarily store stuff on it. And we transport it on those vacuoles. It can be a food one that you're storing a food inside. It can be water, you're storing water, and this can be used for regulation of the osmotic pressure. So food vacuoles, water vacuoles. Food vacuoles, you keep it there until you need it. Water vacuoles are there until you need it whether you need the water or you need to regulate the osmotic pressure, like it's getting too concentrated. Get some water from the vacuoles, basically. Mitochondria, just like ours, to make energy, and they have different types of cycles. Krebs cycle, ETC, or electron transport chain, oxidative phosphorylation. All of this is just like our mitochondria. Chromoplast is another thing that we don't have. In our human cells, we don't have the chromoplast. And not all eukaryotes have it. Only a special type that have the chromoplast. Because chromoplast is actually in plants. The photosynthesis, do we know the photosynthesis, right? If it is during light, during sun time, they use that energy to produce nutrients or to make nutrients, right? Chromoplast or chloroplast. So they take the sunlight, they take the energy and put it into a chemical energy, like they make starch, for example. If you look about any corn, for example, how did they make that corn? Corn is starch, right? They contain starch. How did you make starch? We make it from sunlight. We take that energy and we use it to make starch. So basically, photosynthesis. You're using the light to synthesize the nutrients. Ribosomes, just like ours, protein synthesis. Cytoskeleton, just like ours, but it has a different function. So cytoskeleton is just like ours, microfilaments, microtubules, if you remember those. In our cells, it was more for stabilization, for anchoring the organelles, to keep the shape of the cells, right? This is in human cells. But in other types of eukaryotes that we will talk about, it's different. Besides doing what we just mentioned, it also helps movement. Our cells doesn't move like this, okay? Most of our cells. But this is one of the unique functions. They provide motion. They like contract from this side, they push from this side, changing the shape so they can move. So it is used in movement. So nucleus, simply the manager of the cells. They contain the DNA, they contain the chromosomes. This is where we direct the protein synthesis. This is the manager. They have the original copies, the DNA, from which you can make RNA, messenger RNA, that leave the nucleus, go to the outside, to the ribosomes, to make protein. Do you remember this? That was all physiology. But generally speaking, the nucleus is the largest organelle in eukaryotes. It's the most prominent, the largest, the manager of all, the director of the cell, you can say it. And it's always surrounded by a double membrane. There are two membranes surrounding it. And there are small pores. These pores are small enough that the DNA cannot leave. DNA is bigger than these pores. That's why if you wanted to make protein synthesis, DNA will not be able to go out and tell the ribosomes what to do. And that's why you make messenger RNA that is single-stranded, small enough to pass through the pores. Inside the nucleus, we mentioned the nucleolus. And what's the function of the nucleolus? RNA and ribosomes. Ribosomal RNA and ribosomes. Both of them start with ribosomes. Ribosomal RNA and ribosomes. We know that the ribosomes are actually in the cytoplasm, right? Whether it's free or on the surface of the endoplasmic reticulum. But the question is, where did you make it in the first place? We make it in the nucleolus. This is the assembly site. So if you look at this picture here, you will see a double membrane and small pores. And the nucleolus is inside. The function of each organelle is important. At least for mitochondria, let's say, energy, good enough. For a nucleolus, it is ribosomes and ribosomal RNA specifically. For the nucleus, it's direct protein synthesis. It's the manager, the director of the cell that contain the original copies, which are the DNA or chromosomes. Next is the endoplasmic reticulum. Obviously, endo means inside. Plasmic means the cytoplasm. Reticulum means a network. So endoplasmic reticulum is a network inside the cells. It can be rough, it can be smooth. Rough if it has the ribosomes on the surface, and that's why the surface looks rough. Why does it look rough? Ribosomes stuck on the surface make it looks rough. And obviously those that are rough, because they have ribosomes, and we know that ribosomes is for, what's the function of the ribosomes? Protein synthesis. So obviously the rough ones, since they have the ribosomes on surface, they are responsible for protein synthesis. Why don't you just leave it for ribosomes? Like the ribosomes are free. There are some ribosomes free in the cytoplasm, and some of them are stuck on the rough endoplasmic reticulum. Why don't you just leave it free? There is a difference. The free ones, they make protein and they release it in the cells. So you use it for the cells. The ones on the endoplasmic reticulum, this is a big network. So it's stuck on the surface. In that case, when you make the protein, it will move within that network until you take it to the outside. So obviously it's different, right? It's different, free ribosomes from those on the surface of the rough ER. Smooth ones are the ones that does not have ribosomes on the surface, and this is basically to make and store lipids. Make and store lipids. Isn't it a different function? Rough, you make proteins and transport proteins. Smooth, lipids. You make it and store it. Okay? So if you look at this picture, this is obviously the endoplasmic reticulum, this network, the purple ones. The rough ones are the ones that have the blue on the surface, which is the ribosomes. And obviously in this picture, you're seeing the endoplasmic reticulum, part of it smooth, which is this part here, and the other part is rough, like this, that have the ribosomes on the surface. So the rough ones are the ones that are going to release, to make the protein, and it's going to move within this network, moving, and then they leave from this side right here. Then it will go to the next organelle, which is Golgi apparatus. So this is the difference as I talked about, the free and the endoplasmic reticulum bound ones. How does it move? If you're talking about proteins, it's leaving the endoplasmic, the rough ER, the rough endoplasmic reticulum, and it is going toward the Golgi apparatus. For what? What's the function of Golgi apparatus? Packaging. It's modify also. Modify and package. And modify and package. Like you give me a laptop, it's not ready for exporting. I have to take it, coat it, modify it. I can, if something is missing, I put it, and then I put it inside packages, and then I can export it, right? So you modify. You don't take it as it is, just put it in the packages. Packaging and modification. You modify it. You make it in, looks like the final product that's ready for exporting. So the question is, the proteins leaving the rough ER, moving toward the Golgi apparatus. It moves in the form of vesicles. So you put it like inside a membrane, inside a cyst, inside a vesicle, vacuole, something like vacuoles, and you take it to the Golgi apparatus. So what's the function of Golgi apparatus? Modify, store, and package. Modify, store, and package proteins. And it consists of different cisternae. Cisternae is flat sacs, like you see in the picture there. The brown and or the beige colored, it's sacs like this, folding like this. So the vesicles or the vacuoles entered from one side. Do you have a question?
Yes. So for nucleus that we use, so for Golgi apparatus, would that be what we would draw on for it?
I'm sorry?
For like the nucleus that we have to do, like you said, we have to draw like what it looks like. So would that be like what the Golgi apparatus is?
I don't remember exactly what does it say there, but if so, that will be it.
Okay.
I don't remember exactly what does it say you're talking about the cards?
Yeah.
Does it mention the Golgi apparatus? I don't think so.
Okay.
I think it's just mentioning the bacteria, different types of organisms.
Oh.
One side is organism and the other side is what does it do and the diseases and so on.
Oh.
So I don't think so. I didn't see that at least.
Oh, sorry.
It's okay. So this is the Golgi apparatus. It consists of cisternae. What's a cisternae? Flat sacs. So this is the sequence from the very, very beginning. Nucleus. The nucleus make messenger RNA that goes to the ribosomes. Ribosomes are in the surface, in this case, it's in the surface of the Raphi-R. It makes proteins. Proteins leave the Raphi-R, move toward the Golgi apparatus in the form of vesicles or vacuoles. And then Golgi apparatus is going to release vesicles again for secretion. What's the difference between free and membrane-bound ribosomes? Both of them to make protein. What's the difference? The free ones make proteins for the cell itself, for your own use. But the ones that on the Raphi-R is for exporting. Like, don't you make laptops, for example, to use it in here, and some of them are used to export, right? The one that for your own use are these free ones. Okay. So the vesicles that come from the Raphi-R that contain the proteins, you call it transport vesicles. Okay. The condensing vesicles, on the other hand, these are the ones that take the protein to the organelles or to secretory vesicles, which is moving toward the outside. So we have transport vesicles, condensing vesicles, and secretory vesicles. We have three types of vesicles. Transport vesicles, this is between the Raphi-R and Golgi apparatus. That's it. You just move it from one to the other. Transporting vesicles to transport from the Raphi-R to the Golgi apparatus. That's it. Condensing vesicles will take it to the organelles. Secretory is for outside, for secretion. Here it is. You see these transport vesicles, the purple ones. These are the purple ones that move from the Raphi-R to the maturing surface of the Golgi apparatus. It enters inside the Golgi apparatus, and it is going to leave through the other types of vesicles, depending on what are you going to use it for. Lysosomes. Lyso means lytic, breakdown. Somes means bodies, right? These are the bodies that are used to break down something. Break down means to digest it, right? So how do you digest? It contains enzymes. So if you wanted to destroy something, you send it to the lysosomes. The lysosomes are going to secrete enzymes on it that will break it down, means digest it, right? So we call that intracellular digestion. Do you only digest food? You can digest food, right? If I wanted to break down this food into smaller particles, it's okay. I can send it to the lysosomes. But what if this is an organism or microbe that's invading this eukaryote? The lysosomes can also break it down. The lysosomes, it will break down whatever come to it. Okay, you send them nutrients, it will digest these nutrients. You send them microbes, they will digest the microbes. Foreign bodies, they will digest the foreign bodies, right? So they do not distinguish. They just, whatever you send them, they release enzymes on it. You got the idea? This is what's happening. Whatever you send them, that's okay. They will release enzymes and destroy it. Vacuoles, on the other hand, these are another type of sacs that contain particles that you're sending it somewhere. Like you put it within these sacs and you send it somewhere. You send it to the lysosomes to be digested, maybe. You send it to be excreted outside, to leave the cell membrane and go outside, yes. You just keep it there for some time, storage in case if something happened you can use it. This is another possibility. So vacuoles are basically temporary storage. Temporary storage. You store it, whether because you store it temporarily, and then whatever will be your next step. You can store it temporarily and then digest it. You can store it temporarily and export it outside. Or you just keep it there until you need it. These are the different types of vacuoles, or the different functions of the vacuoles. Yes?
So what's the difference between vacuoles and vesicles?
Not much different, but the vesicles, the vacuoles are not, most of them are not related to the Golgi apparatus. It's just something inside, like a storage inside the cell. The vesicles are related to the Golgi apparatus. Those that take the protein from the endoplasmic reticulum to the Golgi, or take it from the Golgi to the outside or somewhere else. So if it is related to the Golgi apparatus, these are the vesicles. The vacuoles are very similar, but it's storage. You store stuff in it. Vesicles is not basically for storage. It's to transport, to export, and so on. Okay? You're welcome. These vacuoles, those ones specifically, we said that the vacuoles are temporary storage, right? Temporary storage until you do the next step. What's the next step? I don't know, depend on the cell. You can send it to be digested. You can export it outside. You just can keep it there. You can keep it even for months and for a long time. If the condition is not good for the eukaryote or for the eukaryotic cell, you can keep it there for a long time, right? But those who are responsible for digestion, those that will take it to digestion, it will take it obviously to the lysosomes, right? They are going to merge with the lysosomes. We call those the phagosomes. So what are the phagosomes? This is the one type of vacuoles that is for digestion. It goes like this. Okay, here is one type. You have three different types, right? This one is for digestion. This one is for temporary storage. This one is for exporting. All of these are vacuoles, right? This one only, that's responsible for digestion. How does it digest? It moves toward the lysosomes, and then it will open its contents like this. The contents go to the lysosome, they merge together. And the contents go to the lysosomes. Lysosomes are going to throw enzymes on it, breaking it down. You call this one only, special type of vacuole, phagosomes. Okay? Phago means to eat. Phagosomes are and eating we mean digestion, right? You eat something, you're going to digest it. So we call those phagosomes. Next is mitochondria, simply ATP, energy. You make and store ATP. Make and store energy. Make and store ATP. This is a mitochondria. Chloroplast. Chloroplast is something unique in some eukaryotes. Do we have that in humans and animals? We don't. This is basically in plants, right? But there are some eukaryotes that act like a plant, and we will discuss the details. Those ones that act like a plant, they work like a plant, they have that green stack inside that they do not need somebody else to survive. They do not need to be a parasite. They don't have to invade our cells or something. They can work on themselves. They just can take that They can do take the light and they convert it into chemical energy. What do we call this process? When you take the light? Yes, photosynthesis. So these are the ones that can do photosynthesis. And this is in algae and plants. Okay, so plants, we all know plants. Plants have chloroplasts, right? We all know that. That's why plants are always green, right? At least in the beginning, they are green. The leaves are green, right? Why is it green? Because this is the color of the chloroplast. It's green in color. So this is a plant, but the algae is acting like. It's not a plant, but it acts like a plant. In which aspect? Photosynthesis. Ribosomes, we have two types as I mentioned, free and those who are stuck on the surface of the endoplasmic reticulum making it rough. In both cases it makes protein. The difference is the free is for what? The free ribosomes make protein for what? For the cell use itself. You use it inside. While the other ones on the rough endoplasmic reticulum, mostly for export. Okay? But both of them, they make proteins anyway. And these ribosomes, when we talked about the prokaryotes, remember the prokaryotes also have organelles, right? Not all of this, but they do have some organelles. These ribosomes of eukaryotes, they do the same function in a bacteria, but it's just bigger. If you compare ribosome to ribosome between eukaryotes and bacteria, it's bigger in eukaryotes. Internal structure, this is a framework of proteins, microfilaments, and microtubules, and we talked about this in the beginning. It's just like our cytoskeleton. Cyto means cell, skeleton means skeleton, right? Like our skeleton, which is the bone, they have a skeleton inside the cell as well, the skeleton of the cell. These are tiny structures, microfilaments and microtubules. And in ours, it's to keep the shape, to stabilize the cell, right? And also to transport. But in other types of eukaryotes, like protozoa, for example, we will talk about those. But the additional function here will be movement. They can help you to move. Which is called the amoeboid movement or the pseudopodia. Did we hear about the pseudopodia? So it's like if this is a round structure like this, the cytoskeleton is going to push from this side and push from this side. You start to move and you pull from this side. So it helps you to move around in certain types only. So can you answer this question? Packaging, transporting, Golgi. Okay, now we will go to the specifics. What are the eukaryotes that we need to know? Fungus, algae, protozoa, and parasites. And we will need to know details about each one of those. Fungus, algae, protozoa, and parasites. These are all eukaryotic organisms, microorganisms. So we'll start with the fungus. The fungus is usually unicellular. What's uni means? One cell. But they can also live in colonies. Okay, a group of these that lives together. The fungi can be one of two groups, macro and micro. What's macro means? Big. The big ones are the ones that we can see with our eyes. I think we all saw like the mushroom, right? Mushroom and anything that you can see with your eyes big enough to see, we call that the macroscopic ones. The macroscopic ones. Mushroom, the puff balls, and so on. These are all something that you can see with your eyes. Microscopic ones are the ones that you cannot see with your eyes, but remember something. You can't see this one fungus, but you can see the colony. Okay? But I'm talking about the fungus itself. If you look at one fungus, you can't see it. But if you look at a colony, it's different. Like if you look at a bread, don't you look at the bread and say, this is bad, right? This is microscopic. How? Because this is not one, this is a colony. So we're talking about two different things, okay? So one to one fungus and one fungus. Macroscopic, big enough to see. Micro, you can't see, unless it is in a colony. The micro ones are the ones that we care about more, not the macro, the micro, the microscopic fungi. These are the ones that we are going to discuss in details. These are the ones that are clinically important, okay? So how do we classify the fungi? This is important to remember because each one of the 4 eukaryotes that we talked about is going to be divided according to something different. Okay? Are we following so far? So how do you divide the fungi? Morphology. What's morphology? Shape. Morphology means shape. Okay? Yes. So let's look at this one fungus. It looks like a long filament. We call it hyphae. If it looks like a long filament, we call it hyphae. Long filament is also called mold. You need to remember this. Long filament or mold. Long filament or mold. We call it hyphae. This is the morphological categories. If it is round or ovoid shaped, we call it yeast. And the yeast will always reproduce asexually. You don't have to have a male and female to produce it. It just, part of it will detach. Part of it will duplicate itself, just like cells. So this is the yeast. So hyphae looks like long filament or mold. Mold means long filament, okay? We call that hyphae. What if it is round or ovoid shaped? You call that yeast. And does yeast reproduce sexually or asexually? Asexually. Okay. So hyphae, which is the molds, the filamentous ones, if that in the form of mycelia, what's mycelia? Mycelia means a colony of these. It will give you, this is exactly what I was talking about. This will give you a macroscopic, you can see it with your eyes, but you're not looking at a fungus. I'm looking at a mycelia. Mycelia means colony, right? I'm looking at a whole colony. Yes, like look at these pictures here, you can see it. But this is still microscopic. You're seeing a whole colony with different colors, whatever you see it. White, red, green, whatever it is, you're seeing the spores. Okay, like what we see in the bread, for example. On the bread, it's actually a very tiny, it's microscopic ones, but you're looking at a colony, mycelia. So if that's the case, each group of mycelia, each group or each mycelia or each colony can give you a different color depending on the spores that in that colony. Okay, did you get my idea? You're looking at it and you can see it with your eyes. Can I say macroscopic? No, it is not. It is still microscopic. It's still mold. It's still hyphae, but you're seeing a whole colony. Okay. The next type is the yeast. And is the yeast filamentous shaped? No, it is round or oval or ovoid, like kind of oval. Okay. Of course, it is asexual. The way they divide is usually by budding. When you see buds, remember yeast. It's asexual, right? It's asexual anyway. But what kind of asexual? Budding. When you see budding, you're talking about not just fungus, you're talking about yeast. Look at this bud. You just make a protrusion like this, a bud, and this bud will separate. Now you have two, right? And each one of those two will make a small bud, and this bud will become bigger and detach. You now have 4 and so on. So this is how they reproduce. Buds. When you see bud, Yeast. Bud, yeast. Because it's eukaryote, they have all organelles, but the yeast have also a cell wall. Not all eukaryotes have a cell wall, right? Actually, most of the eukaryotes do not have a cell wall, right? We have a cell membrane. Do you remember this? Prokaryotes, they do. Eukaryotes, yeast have it. The yeast have a cell wall. And they have all types of organelles. The one that they do not have is the locomotor organelles. So they can't move. Like flagella, do you remember these? Flagella, cilia, and all that, they don't have it. Other than that, they have everything else. Mitochondria and ribosomes and RAF-AR and smooth AR, everything else. They have it, not locomotor. Is that clear? Yeast cannot move, remember it. Yeast, generally speaking, if you're talking about one of those, it's bigger than the bacteria. There is some types of bacteria, not bacteria, fungus I mean, that can change the shape from yeast to hyphae or hyphae to yeast, and you call that dimorphic. What's di means? Two. And what's morphic? Shape or form, yes. So dimorphic means two shapes, right? It changed from filaments to round structure or yeast, or from yeast to filaments, from yeast to mold, from mold to yeast. Is that clear? So if that's the case, you call it dimorphic. So what's the difference between yeast, mold, and mushroom? Yeast is basically a single-celled fungus. There is one type that's important for humans, and this one is called the Saccharomyces cerevisiae. Saccharomyces cerevisiae. You know what's Saccharomyces cerevisiae? This is the yeast that we buy in the store. You go buy a yeast and you use it like to make any form of foods or something, right? We buy the yeast. So we're talking about this one. Saccharomyces cerevisiae. Okay? They use it to make food. They use it to make alcohol. It's a yeast. Okay? And even in the store, it's called yeast, right? Mold, on the other hand, they grow in hyphae, and if you look at this picture here, looks like this. And mold is basically the one that is bad. Like when you buy a house, don't they tell you like look for molds? Do you remember this? Like it can come like in the woods, in the basement, something like that, right? It grow in spots that you hate to see. These are mostly the molds will be like this. Unlike the yeast is beneficial, right? We use the yeast. Alcohol, bread, right? Food in general, it's used. Yeast. The molds, when you see it, it's usually something bad. And it looks like this. If you ever see something like that before, inspecting a house or something, you see something like this, this is a mold. Mushroom, on the other hand, this is the shape that you can see here, which is something that looks like this. The classic one that we sometimes see in the backyard or something, it looks like this. Nutrition. All fungi are heterotrophic. What's hetero means? Huh? Different, yes. What's trophic means? Feed. It's more of a feeding, or how they eat. Okay? Heterotrophic means they feed on something else. They do not feed themselves, meaning, right? So obviously, do they have chloroplast? Of course not. If they have chloroplast, they will not be heterotrophic. All fungi are heterotrophic, no exception. They do not have chloroplast. Okay? They do not have chloroplast. So they cannot live by themselves. It's not homotrophic. Did you get the idea? It's heterotrophic. So they need to live on something else, not necessarily in our bodies or animal bodies. They can live on anything, even dead plants or something which is not bad, right? It's not harmful. So the majority of these fungi are harmless. Some of them are harmful. The harmless ones are the ones that are actually beneficial. It's harmless and sometimes it's even beneficial, like saprobes. Saprobes is a name of the fungus that's responsible for decomposition. Do you remember decomposition? When something is there, dead plants, dead animals, and then they decompose, right? Disappear, basically. It doesn't really disappear, it's just decomposed. And it mixes with soil and so on. And it looks like it's disappearing, it's not. It's just decomposing. And what do you call these harmless fungus? Saprobes. Okay? Saprobes. So what's saprobes? Harmless fungus that does decomposition. Is that clear? Harmless decomposing fungus. Saprobes. On the other hand, there are other types that are harmful. Harmful ones are the ones that live on something that is living tissues, human tissues, animal tissues, right? So this is obviously a parasitic, right? A parasitic. So did you get the idea? If they live on dead, not living, dead, whether it's plant or dead animals, dead anyway, not living, dead. We call that harmless and saprobes. If they live on living tissues, human tissues, animal tissues, we call that parasites. Did you get the difference between those two? Okay. When these types of fungus, which is parasites, when they cause disease, whether it is in animals or humans, we call that mycosis. So, like if you have, we know the athletic foot, right? Athletic feet, isn't that a fungal infection? We call that infection mycosis. Where did you get that word from? Mycos is another name for fungus. Mycos is another name for fungus. So mycosis is you're being invaded by fungus, which is a fungal infection. Okay? So what do you call it if an animal or human is infected by fungus? Mycosis. Mycosis. So if you look at this, this is athlete's foot. See how it looks like? What's the organism causing this? It's mycos, yes, but which type? Does anybody remember? Tinea. Yes, tinea. So it's tinea. I'm just asking. Most likely we forget, of course, but just if you remember. And if you think about athlete or athlete's foot, what's that? Why did you call it athlete foot? Yes, because those athletes, they work for or they play for a long time. They have a lot of sweat. So this humid environment is very encouraging to this type of fungus. Okay. So fungi, rule of fungi, it can be beneficial and it can be bad, right? Some of them are good, some of them are bad. Not all of them are bad. The beneficial or the beneficial one is decomposers. This is something good. There is no need to keep dead animal bodies, right? Or dead plants, what they need for. Actually, when these decomposers, which is called What do you call these decomposers?
Saprobes.
Saprobes. Remember this, saprobes. So these saprobes, which is decomposers, this is actually something good. Because when they decompose the dead animals and the dead plants, they enrich the soil, right? So the soil will be better. Whether we're talking about better for other plants to use these nutrients or to enrich the soil, oil, it can make it in millions of years and so on. The other thing that's beneficial is, beside the decomposers, is they can use it to produce antibiotics. They take it in the lab and they do some work. They invade them with something until they get the antibiotics and they use it. Others are for nutrients. They can use it for alcohol, for bread. They make organic acids. They make vitamins out of it. There are a lot of benefits from fungus, beside the genetic studies. So these are all the beneficial ones. The bad ones is that they can infect us, right? What do you call it when they affect humans or animals? Mycosis, yes, mycosis. And the other thing that's bad, they do not even have to invade us. Sometimes we can have allergy against fungus. Just like, some people can have allergy against peanut, for example, right? Some people have allergy against the certain types of fungus. Whenever it's available in the environment, you just smell it and you get that, start to cough and sneeze, and you have these reactions. This is allergy. And some of them are even produce toxins. They are toxic. Of course, all of this is bad. The other thing is they can invade our plants, the crops, the food storage, right? Like the moldy rye, they are storing the corn, and then you look at it and you see these fungus on it, you throw it away, right? Isn't that bad? This is the bad part. Adverse impact. So all of these, invading us, mycosis. We produce allergy toward these, it's allergic reaction. Some of them produce toxins. Some of them invade our crops and food storage. Okay? Mycosis. If we're talking about mycosis specifically, which is fungal infection of humans, right? The vast majority of the fungus is superficial. This is what we hear about all the time, right? Athlete's foot, you have something on your body from out, it's always on the skin, right? This is the superficial form, which is the vast majority. We call it superficial. They do not invade, this is what we actually familiar with. This is what we can usually see, which is superficial, like it can be on the outer epidermis only, like tinea versicolor, for example. It can be epidermis but invade some of the derms and hair, like tinea. So tinea does not only involve the epidermis, it goes a little bit more. It goes to the derms and to the hair. Okay? Like tinea, we talked about this. Ringworm, athlete's foot, all of this. It's still superficial, but not only epidermis. Tinea versicolor is only epidermis. Some of them invade even to the mucous membranes. Candidiasis is the most common one that we need to remember. Candida albicans. What albicans mean? White, yes. White. Candida albicans, why did you call it this? It's Candida. This is the name of the fungus. And albicans because if you are invaded with those, you will see the inside of the cheeks that's looking white, and some spots are red. White and red. So if you look at, if this patient, you open the mouth wide and you look inside and you see the mucus. Mucus is the inside of the cheeks, right? So if you look at the inside of the cheeks, the mucus, and you see white patches like this, and you see some red on it, this is definitely Candida, candidiasis. He or she are invaded with the Candida. Okay? Of course, they do not take it like this. Not like open your mouth, you have Candida. They take a swab, they look under the microscope, and guess what? They have a unicellular organism there. They look at it. It has a nucleus, looks like eukaryotes, right? It might be dividing, asexual, and so on. They look at it under the microscope to know exactly what it is. Okay? On the other hand, there is systemic ones. Systemic means it goes inside to the systems, not just the skin. Okay? Like lung is the most common one. They can invade the lung. They can also invade the lung and the skin at the same time. Let me say this. If it lung and skin, this is also kind of common a little bit, like histoplasmosis, for example. Histoplasmosis. Cryptococcosis, another example. Coccidioidomyces, plastomycosis. These diseases, histoplasmosis, let's take it as an example. I mentioned that usually the fungus is superficial, right? 90% of the time, if you have a fungus, I can tell you it's invading your skin or mucous membrane, right? And this is superficial. It does not invade systemically. It doesn't go to the systems, except these kind of exceptions. Histoplasmosis, for example, this is more common in states like Arizona, where it have a lot of dust, right? So it can be like in the dust and you're breathing that, it go to your lung and invade you, okay? How can it invade you if you are healthy? And it's usually invade the skin. This is an exception. Some of them, just you inhale them. And this is, again, it's not widespread. If you are invaded with a different type that goes inside, but not like this. It goes inside your lung just because you're breathing it, right? It finds a route. But how can it go like to your blood and to the organs inside? If that's the case, you are immunocompromised. Okay? So if you are healthy, let's say it like this. You have a fungal infection. Are you healthy or immunocompromised? If you're healthy, it's usually superficial, which is the skin or mucous membrane. Okay? Or occasionally it can be the lung, like histoplasmosis. All right, so far? If you are not healthy, if you are immunocompromised, it can cause systemic. So it's completely not normal to see a healthy person with systemic mycosis. Is that clear? The only exception is this one, because you breathe it, which is something like histoplasmosis. How to identify fungus? I mentioned it. You see a patient, open your mouth, you see white patch and some red areas, and you say to yourself, okay, this looks like Candida, right? Candidiasis, Candida albicans. This is not enough to look at it. You need to take a swab and put it in a specific media. We call that isolation, inoculation in the media. And then you look at structures that are asexual spore forming. Look under the microscope, they make spores. Okay? If it is hyphae, what kind of hyphae? If it makes colony, what kind of pigment? Is it making green, yellow, red pigment? Depending on that pigment color. Physiological characteristics. How do you can study everything about them. You can even do genetic map makeup. If that's the most advanced. Like if you study the genes, you know for sure what it is. But you can tell even without the genetic makeup with all of these. Let me see what type of hyphae you have. Let me see the spores, the spores that are asexual, how the colony looks like. You can tell from these. But the most advanced is genes, genetics. If you look at the genetics, you can tell. So can you tell me which of these can be used for classification that we just mentioned? B and C, which is E, right? Okay. So this was the fungus. We're done with the fungus, okay? Any questions about the fungus? We're good so far. Okay. So the next eukaryote is protists. Protists are organisms that they put it in a separate category because they do not fit anything else, basically. It's not animal, it's not plant, it's not fungus. What else is it? We just put it together and we call them protists. Okay? And protist can be one out of the following. Can be algae or protozoa. Algae and protozoa, it's not a fungus, but it's not a plant or animal. What else? We call it protist. Okay? Are we good so far? Okay, so obviously both algae and protozoa, both of these are eukaryotic organisms. Okay? Usually it's unicellular, sometimes it's colony, but they are unicellular. The algae is a protist that's closer to plants. Protozoa is a protist that's closer to animals. Okay? It's not a plant, it's not an animal. Are we clear? We put it in a separate category because it's not a plant, it's not an animal, and it's not even a fungus. It's a protist, right? But what kind of protist? If it is closer to the plants, it's not a plant, but closer to the plant, in which way? Chlorophyll. They have chloroplast, right? Chlorophyll. So they can do photosynthesis, right? So it's unicellular most of the time, sometimes colonies, but most of the time unicellular, and they can do photosynthesis. You call that algae. So is that algae homotrophic or heterotrophic? Homotrophic, right? The algae is homotrophic. If you leave it alone without anything else, can it survive? Of course, yes. They can just sunlight, convert it, use it, and that's it. Photosynthesis. If you can do photosynthesis, you can live on your own, which is called the homotrophs. Protozoa, on the other hand, is also unicellular, and they have a lot of similarities, but it's closer to animals. Okay? And obviously these are heterotrophs. So algae, most of them are free living in fresh and marine water, like planktons. We call it planktons. Not only that they feed themselves, but they are actually making food web to other aquatic habitants, something like fish. They can feed on those, okay? Or any other organisms, any other habitats in the water. They can use these algae as a source, just like humans. We use plants, right? Plants are homotrophs, right? They have chloroplast. They can live on their own, and we use them. Algae do the same rule, but in the water. Okay, this is the food or the food web in the water. We also use it to make agar. Do you remember the agar? The algae is used for red algae is used for agar. Also, just like plants, they take carbon dioxide. You guys did biology before, right? They take carbon dioxide, they take the sunlight, they put them together with water, and this is how they make nutrients, right? Carbon dioxide, water, and energy from sunlight, right? This is how they make. So basically they are getting rid of carbon dioxide and they give us oxygen. So this is the source of atmospheric oxygen. Not the only source. Plants is the major source, right? But these also provide some source. If you look at this picture right here, sometimes you see this red tides on the surface of the water. This is called dinoflagellates. Looks like this picture here. So remember this name. If you see the red tides, look at the water and you see it getting reddish. You see tides that are getting reddish, and you're wondering, what is this? This is a type of a protist that's called algae, and it's which algae specifically? Dinoflagellates. We're not going deeper in the algae, but this type is important to remember. Dinoflagellates. Dinoflagellates like this, what you're seeing there, and this is basically, they give that red tide look to the water, and if that's the case, you're not supposed to go closer to that water. Okay? If you see the red tide, stay away. Why? Because they produce toxins, and this toxin can make neurological symptoms. So if you go swimming in these When you see the red tide, stay away. If you go there, you can get poisoned, you can get these toxins, and they bad enough that they can do like neurological symptoms. They invade, the toxins can be harmful to our nervous system. So this is the algae. See, it's green because they have the chloroplast or the chlorophyll. This is the algae cells itself. Next is the protozoa. Protozoa do not have a cell wall. Do not have a cell wall. Okay? Usually unicellular. Most of the protozoa, just like the fungus, are harmless. Does the fungus have a cell wall? The fungus does have a cell wall. Protozoa does not. Okay? Protozoa does not have a cell wall. Most of these are harmless. If it is harmful, we call it parasite. Just like the fungus, right? Parasite. If it is harmful, we call it parasite. Like those that can invade us through an insect vector. Like malaria, for example. Do you know malaria? How does it come to us? Mosquitoes, right? Mosquitoes. So, which is an insect, mosquito, and it lives in the mosquito, and it can get into our blood. Malaria is an example of parasitic one. All of these protozoa are heterotrophic. Heterotrophic. Do they have chloroplast or chlorophyll? They do not have chloroplast or chlorophyll. So they have to be heterotrophic. Are the algae heterotroph or homotroph? Homo. The algae, they can live. They have their chlorophyll. Their cytoplasm, which is different than other types of eukaryotes, it's actually ecto and endo. What's ecto mean? Outside, yes. And endo, inside. So it's two components. Instead of just being a cytoplasm, just like this, it's part of it is outside and part of it is inside. It's like two components. They engulf microbes or organic matters. They engulf it, they eat it, and this is how they get their food. Obviously, they cannot live on their own, right? It's heterotroph. So whatever they can eat, they eat it. Whether it's something organic material, organic matter, microbes, whatever it is. Protozoa usually have locomotor structures. Locomotor structures. Remember the fungus, the yeast, does not have a locomotor structures. Remember we said all organelles except locomotor. Protozoa, they do have the locomotor structures. Whether we're talking about flagella or cilia or pseudopods, they have something to help them to move, most of them. Okay? Are we okay so far? They exist in different forms. There are two forms that we need to remember specifically. And the two forms that we always see, which is either the trophozoite or sporozoite. What does that mean? Tropho, coming from trophic. These are the ones that are the vegetative form. These are the ones that can eat. Okay? These are the motile ones, the vegetative ones. These are the ones that actually moves and eats. Trophozoite. Sporozoite means they live temporarily, in the form of spores. Do you remember the spores? When they get smaller, shrink, and have vacuoles of food, and they have a lot of layers to protect them, right? So sometimes if the condition is unfavorable, like they don't see nutrients, it's getting too cold, too hot, something like that. It's not favorable anyway. If that's the case, they become dormant. Is that vegetative? Sporozoite is not vegetative. It's a spore form. Spore form is like this. They shrink, they acquire more cell wall and stay like this. Don't do anything. Until the condition improve, and then they return back to the trophozoite. Did you get the difference? Trophozoite versus sporozoite. Vegetative versus spores. Dormant. Tropho means trophic. These are the vegetative form. These are the ones that actually survive, they live, they move, they eat. The vegetative form. Tropho. Trophic means to feed. Sporozoite is spores. Spores are dormant. Okay? They don't do anything. Just stay until it gets better. So if you look at this picture here, here's the trophozoite. Acquire a wall. They become mature. They stay. If condition gets better, they change and become trophozoite. Okay? The way we classify protozoa, do you remember how did we classify fungus? Morphology and asexual reproduction, right? This the protozoa are classified according to locomotion. How do you move? Are you using flagella? Are you producing pseudopods? Are you using cilia? Or are you non-motile completely? You do not move. Most of them they do, okay? But some of them do not move. So we said that a lot of protozoa are not pathogenic, right? They are not harmful. Let's talk about the harmful ones. The one that can actually cause disease, that can invade us. We call those pathogenic. Okay? Pathogenic. The first type that use flagella. Example, trypanosoma. Trypanosoma is an example of these that use flagella. Okay? Trypanosoma, Brucei versus Cruzii or Cruzii. Brucei is the one that cause sleeping sickness, African sleeping sickness. The Cruzii one is the one that cause Chagas disease. They call it the kissing disease. Obviously, it moves through or infects through kissing. The sleeping sickness, they found it's more in Africa, but it can be in any hot environment. And when somebody's invaded with this type of protozoa, which is trypanosoma, they are sleeping all the time. Like they go to the doctor, sleeping 15 hours a day or something, and they start to have like something in their skin and they look different, but the main thing is they are sleeping the whole time. And then they get a sample from their blood and they look until they found that trypanosoma. Okay? Trypanosoma is transmitted by blood-sucking vectors, insect or something like that. Whatever, any type of insect, whatever vectors that can suck blood. Okay, they can take it from one person. They suck the blood from one person, including these trypanosoma, and then they go inoculate that in another person's blood. So this is how it's transmitted. Are we okay so far? And how do they move? They use flagella. Flagella. Trypanosoma use flagella. And they're transmitted by blood-sucking vectors. Trypanosoma. Clear? Trypanosoma. They move by flagella. Transmitted by blood-sucking vectors, whatever the vector is. Next time. Remember that we're classifying according to what? The way they move, which is locomotion, right? The way they move. The first type moved by flagella, which is trypanosoma. How do they invade or how do you get infected? Blood-sucking vector. A vector that sucks blood from you and give it to somebody else. Clear? This is what we need to know about trypanosoma. Second type, it uses pseudopodia, or they call it, they have the amoebic type of motion. They do not have a flagella, they do not have a cilia or anything. It's their own bodies. They don't have appendages. Their own body changed the shape to make something looks like feet, and we call it pseudopodia. What pseudo means? Fake or false. Yes. So they have the fake or false feet, and that's why they call it pseudopodia. So if you're using pseudopodia, what do we call this movement? Amoebic or amoeboid movement. Amebia-like movement. Entamoeba histolytica. So entamoeba, like Entamoeba histolytica, for example, they move around like this. Let's say this is the entamoeba, something like this. They start to move and they do something like this. Okay, start to extend. So it looks like a feet, and start to like move around. And they change the shape and they move like this. If you want to move this way, they make the feet this way, and so on. Okay, so they use pseudopodia, and we call this amoeboid movement. When you use the pseudopodia, we call it amoeboid movement. Okay? How do you get Entamoeba histolytica? You get it by ingesting cysts. Okay? This is the one thing that if you go like overseas or something, they tell you, do not drink from any water, right? You have to get bottles or something like that. Otherwise, you will get the amoeba. This is what they're talking about. Okay, they advise you. If you go there, drink from bottled water, right? Do not drink from any other sources of water or any other juice or something like that. Try to avoid that. They are feeding this because sometimes the cyst will be in a food or drink or something, and you ingest it, and it go to your small intestine specifically. Is that okay? So how do we get let's say from the beginning, we are dividing them according to their locomotion or movement, right? This type use, the entamoeba use what? Pseudopodia. They use pseudopodia to move. Okay? What else do we need to know about this infective amoeba, which is Entamoeba histolytica? Beside using the pseudopodia, and what do you call it if you're using pseudopodia? Amoeboid movement. Amoeboid movement. Okay? So if they're using this pseudopodia, what else do we need to know about them? How can we get infected with those? Cysts in food or drink. You're drinking or eating something that have the cyst. It's not clean enough. Sometimes you get it even in the food, like salad or something, like this person didn't wash his hands before cooking or before preparing salad or something, so he have these cysts, and you eat the salad or drink water that have the cyst, it go to your small intestine, it hatches, the cyst will hatch, and the entamoeba itself is going to invade your intestine. In the beginning, this is extras, you don't have to know it, but they call something called amoebic dysentery. Like sometimes we hear it a lot, like somebody's coming from overseas and say, I have like some troubles in my stomach, when I go to the bathroom, I can't make it, it's like constipation, but not really constipation, like a diarrhea or constipation. You see something like this. This is amoebic dysentery, and this is still in the beginning. If this person is weak enough, they are going to invade other organs. Okay? Most of the time they do not. But they can invade other, like liver and lung, if this person is very weak. Next type is called, and again we're talking about how they move, right? They have the next type that move using cilia. Cilia. They have cilia, which is hair-like structures, and this is how they move. And the example of that is Plantidium coli. This is something that lives in the colon, and this is where the name came from, Plantidium coli. And just remember that it moves by cilia. Okay? Next type does not move. They do not have any of those. No cilia, no flagella, no pseudopodia. An example of that is sporozoa, like Plasmodium species. You do not have any of those. They just move with the bloodstream, okay? And the best example of that is Plasmodium that cause malaria, okay? Plasmodium vivax, or whatever the Plasmodium is. Plasmodium is a species, by the way. So what's a Plasmodium? This is a sporozoa that does not have any mean of movement, okay? We call that Plasmodium. Plasmodium what? Different species. We're not getting into the details now. Just know for generally speaking, it's a Plasmodium sporozoa, does not have a mean of movement. Clear? Like malaria. If you ask yourself how malaria moves, it just moves the bloodstream. Okay? They do not have their own means. Last one that also use the flagella is Giardia lamblia. They have a whip-like structures. Look at these. They have these long flagella. They use it like looks like a whip-like structures, and this is how they move. It also invade the intestine. So which types use flagella? Trypanosoma and Giardia. Both of them use flagella. It's different, but they use flagella anyway. Which one does not move? That does not have a mean of movement? Plasmodium. Which one uses the cilia? Plantigium. Plantigium coli. Plantigium coli. And which one uses the pseudopodia? The amoeba or the entamoeba histologica, as an example. Which of those? Algae and protozoa. Always? Is it always? We didn't say always, right? It's mostly. Are both of them photosynthetic? Which one is photosynthetic? Algae only, so this is not right. Both of them have mitochondria? Every single organism have mitochondria. All these eukaryotes have mitochondria. So yeah, that's true. Is both of them heterotroph? Which one is heterotroph? Protozoa, right? So by exclusion, and it's not by exclusion, we know that all eukaryotes have mitochondria anyway, right? This is where they make the ATP. Okay. Next type is helminthes. Helminthes means worms. Okay? So parasitic helminthes are the worms that invade us. They are parasitic. Obviously, this is never unicellular. It's a whole worm, right? So it's multicellular. And they have organ systems. They have reproductive system, digestive system, skeletal system, and so on. Some of these are parasitic, which is what we're talking about. The parasitic ones are the ones that have a host. They live in a host tissue. Okay? Are we following so far? These parasites, they have a mouth part to help them to attach to the digestive system, which is called scolex. Are we following? They have to have it. Otherwise, if they don't have it, they go to our intestine and they go in a feces, which doesn't happen actually, right? If you ask yourself, like you're ingesting something with these worms, okay, you ingest it, it leaves. No, it doesn't happen because of scolex. What's a scolex? Mouth part for attachment. For attachment, it goes like this. You have a mouth like this with something. You have something looks like teeth, okay? And they go to the intestine and they attach themselves, and that's why it doesn't leave. It goes in and it doesn't leave. They are using the scolex. They have well developed 6 organs. So this is not a sexual reproduction, right? This is asexual reproduction. This is sexual reproduction. They produce eggs and sperms, which is used for reproduction. Okay? So there is male and female, right? Worms have male and female. Okay? The male produce sperm, the female, just like animals, humans, right? Are we following so far? Okay, so they do produce eggs and sperms. There are male and females. However, there are some types of worms that's called hermaphrodite. What's hermaphrodite? Both of them. They are male and female at the same time. They produce sperm and eggs. They fertilize itself. So we call that hermaphrodite. When they produce the egg and the egg is fertilized, so the sperm came to the egg, it becomes fertilized. Now it will become larva. Okay? So these eggs that will become larva, this is how they get in and out of the host. They get into the host like an egg or larva, and they leave in that same form. Okay? Some of them will invade you by being an egg, fertilized egg. Some of them will get to you being a larva, and they leave in the form of eggs. Okay? So they alternate the development between larva and eggs. Eggs become larva. When the eggs hatch, it become larva. And most of the time they have alternate host. What's alternate host? Like if they invade us, humans, they invade something else also. Okay, like the fluke worms, bilharziasis. They invade us, okay? When they leave our body through urine or feces, it goes into the water and they live in another host. And this host is snails, for example. Isn't that another host? So you usually have more than one host. They stay in the snails for some time, they leave the snails, go to the water, invade us again. So we are hosts, snails are another host. Okay? So they alternate hosts. So these are different types of helminthes, but we have some details. But look at this. Look at this one here. What is this? This is the scolex, which is a mouth part for attachment. Otherwise, it cannot infect us, right? How can it infect? They infect because of this. We have different types of parasitic helminthes depending on how do they look like. Is it flat? If they look like flat, meaning they do not have cavity, just flat, we call that the flatworms. If it is not flat and they do have a cavity, it will be the roundworms, which is common. Okay? Flatworms. They do not have a well-defined digestive system, these flatworms, because why? They are flat. They don't have cavity. They cannot have a well-defined digestive system. They have a blind-ended pouch. That's it. Just a pouch that blind-ended, because something come in and out. Okay? They just have a simple excretory and nervous system. Just a little bit of those. What are the types of flatworms that we need to know? Tapeworm is one thing. Flukes or fluke worms are another thing. So what's up the flatworms? Flatworms are worms that are flat. What do you mean by flat? Do not have a cavity. So what? So they do not have a well-defined digestive system. Okay, so far? They have blind end. This is subdivided. The flatworms subtypes are tapeworms and flukes. Tapeworms looks like tape, basically, and you call it cystodes. You need to remember both names. Tapeworms are called cystodes. Flukes are called trematodes. You need to remember this. Okay? Tapeworm, cystodes. Flukes, trematodes. Okay? So the tapeworms or cystodes, this is long, flat, and they form of repeated segments of something called proglotidus, which is this. Okay? It's a series of these pods. Proglotidus, one after one after one after one. Okay? These are the components, these are the segments. They do not have a digestive system. And these tapeworms usually come from undercooked meat. Okay? Basically, they live in the intestine of vertebrates. We know the vertebrates, right? Anything that have a vertebral column. Animals, basically, animals that have vertebra. And how do we get infected? We get these animals, we eat the meat that's not, some of us like to eat the meat that's not well done, right? Kind of raw a little bit or half. It doesn't mean that if you eat it like this, you'll get infected, but you have a chance, right? If this animal was infected, undercooked meat, and they live in the intestine, basically. Flukes, which is the trematode, so this is the first one, okay? This, the tapeworm or the cystode. The second form, which is this, are flukes or trematodes. They are also flat but not segmented. They have a sucking mouth part. Okay? So which one is segmented? The tapeworm. The cystode is segmented. And what do you call these segments? Proglottids. Proglottids. These are the segments. Fluke worms are not segmented. It's just one piece. Okay? And they have this mouth part. The other type that's not flat. So the first one is flatworms. Flat. Whether it is a tapeworm, cystode, or flukes, trematode. Okay, so far? What if it's not flat? It's round. We call it nematode. You need to remember both names. Round is called nematode. So this is round, obviously, and because they are round, it means they have a cavity. Since they have a cavity, they have a complete digestive system, right? This is usually found in the digestive system or in the blood. And this is how it looks like, round. How to identify the different helminthes? Well, it depends on the stage. Are we talking about eggs, larvae, or a whole worm, right? We have different forms. Egg, larva, worm. Egg, larva, worm, right? Egg become larva, larva grow and become worm, right? Egg hatches into larva. Larva grow, become mature, which is the mature worm, right? If it is an actual worm, or even if it is a larva or egg, look at the shape. Obviously they look different, right? Is it round? Is it flat? Is it segmented? Is it not segmented? Size, is it big ones? Small ones? Organs, like round have digestive, right? A complete digestive system. The flatworms does not have it. Do they have hooks and suckers or not? So you can classify according to that. How do they reproduce? Is it hermaphrodite? Is it male and female? So we use all these characteristics and host when they invade us. Do they go to the intestine? Do they go to the blood? Do they go to the muscles or what? Okay? And how did the eggs or larva, if you're talking about the eggs or larva, how do they look like? So if it is worm, how does it look like? Larva, how does it look like? There are different species of worms or parasites. There are a lot of species, a lot of different types. Some of them are all over the world, just distributed, and some of them have geographic distribution in certain areas only. Usually you get infected either by the eggs or the larva. Okay? You can get it in the food. You can get it in the soil. You can get it in the water. You can get it from a vector, insect vector, but never through air. Did you ever see a worm like flying in the air or something? Never in the air, right? It can be in the water. You drink, not necessarily you drink, by the way. Schistosomiasis, for example, schistosomiasis, which is called bilharziasis. The way they infect us is you go to that stagnant water, you put your feet in or your hand in, and this larva have a very thin mouth and it's very small. They penetrate the skin and goes inside until they reach the intestine or the blood. Okay, so water not necessarily drinking. It can invade. You put your feet in the water, it invade your skin. It's still water, right? So they live in the water. They live in the soil. They live in the muscles or the intestine of animals. So you get it through food, water, soil, or a vector, never air. Okay? This is their life cycle. Egg, larva, worm. Egg, larva, worm. If you look at this picture there, male and female, they produce egg, the egg become larva, and it can be transported or it can infect in different ways. You can infect yourself, and we call that auto-inoculation or auto-infection. Auto means self, right? You infect yourself. Most of these worms, like this is the pinworm, okay? Why did you call it the pinworm? Because it literally looks like a pin, okay? Very thin, you know how the pin looks like? Looks like pin. Very tiny ones. So the life cycle will come, you have a male and female, they lay eggs, right? The eggs will hatch and become larvae. And you either auto-inoculate yourself. Some people, we said that these lives in the intestine, right? They live in the intestine. So pinworms specifically, look at this picture, they did it like this for a reason. These kids, specifically in kids, because kids eat anything, right? They eat or drink anything. So in kids, here is a kid, here is a child who's saying the following, okay? And I've heard that a lot. They say, I'm fine all day long, but I wake up in the night time and I scratch this buttocks around the anus. They scratch very bad. It's really bad that they have to do this, and then it disappears. What's happening is these pinworms, they live in the intestine. Okay, they have this habit that they leave and go outside through the anus and go around, and they start to use their tip in the skin, and they produce this painful, itchy. So that's why they wake up like it's bad. They itch. We know exactly, once you hear this, it's a pinworm immediately. There's nothing else that does this very characteristic kind of infection. Wake up in the night time, scratching around this area in the anus. What it is, it's a pinworm immediately. Okay? They leave at night time specifically. They start to pin. They start to make these sucking blood, and they start to do these forms, and then they leave and go back. They stay for some time and then they go back. This is their complaint. So sometimes people like scratch in this area and then they do not wash and then they eat it again. This is called auto-inoculation or auto-infection, okay? Or sometimes, again, the same thing, you're scratching and you didn't wash and you're touching somebody else and you invade somebody else. And this is called cross-infection. Auto-infection, you infect yourself. Cross-infection, you infect somebody else. Okay? Can you tell me which one of those is true? C. Okay? And that's it for this chapter.